Okay, this is where we left off. As we were going through the first part of the notes, what we were doing is we were looking, after reviewing the basics of learning and memory, we were looking at what's going on in the brain and whether certain brain regions are involved. And we saw what Lashley tried to do, and Lashley was looking for the engram, but ended up not being able to find the engram apparently. And he claims principles like the principle of mass action, the brain works as a whole, and equipotentiality. Every part of the brain does works contributes equally to the forming the memory and functioning and learning and memory. But what we saw by getting into some of the findings related to the medial temporal lobe is that that's really not true. And so we want to continue on with this kind of in this approach here of looking at different brain regions and the roles that they might play in different types of memory functioning. In order to do that, we want to update our ways of thinking about memory to also look at working memory, right? So this is our modern version of short-term memory rather than, the short-term memory idea as was originally framed, especially back in the 1950s and 60s, was a natural outgrowth of Hebb's dual trace hypothesis, the idea that there would be a short duration temporary form of memory and then a more longer duration semi-permanent long-term memory, right? But what Alan Baddeley reasoned in the late 70s into the 80s was this idea that what we usually think of as short-term memory is really this place where we're keeping information active so that we can do something with it, perform mental operations and engage in a task, work with the information and reason with it. So, of course, he says we should call it working memory. But he also notes here that working memory has different forms, that we have different kind of types of memory. We have a visual, spatial kind of memory. And so our spatial reasoning abilities would be distinct from, say, our verbal memories, right? And that's called the phonological loop. So here in the middle of the model on the left, we have the visual spatial sketchpad. That is our visual and spatial information that we use working memory for. Recognizing a familiar visual object, like say a stop sign, for example, has is part of that. The phonological loop, though, is that inner voice, right? When I'm thinking and working through a problem and I might be using language internally to try to reason through to solve a problem, then we can activate, that's our phonological loop. The episodic buffer is our memory for events and episodes that have occurred. These are all interconnected to long-term systems that are also somewhat modality specific as well, right? So a long-term visual memory, a long-term language memory, meaning of words and so forth, as well as our episodic long-term memory, my long-term memory of events and things that have happened in the past. But at the top of the box there is the central executive. So this is what we usually think of as executive function or executive control, the ability to allocate attentional resources to these different systems, to spend more time doing one task as opposed to another, maybe also to allocate attentional resources. Um, we'll call that attentional control. And we could maybe try to map this onto the brain as well. And what we're seeing here is that we're going to see lots of different brain areas involved. It's not just working memory in one location. So this is maybe a little bit like mass action, but don't be misled by that, because what this also shows is the principle of equipotentiality does not really hold to be true here, right? Think about this. So we'll look at the frontal lobe first, right? Prefrontal cortex is where we're going to put our central executive. Other frontal areas, maybe the attentional controller. So the frontal lobe is going to play an important role for this here. When it comes to the phonological loop, though, yes, the frontal lobe is important because we got Broca's area, and that's our inner speech production region of the brain. But we also have Wernicke's area in the temporal lobe, that is the speech comprehension. And this idea that information is looping back and forth between Broca's area and Wernicke's area is one way that we might think about how the phonological loop could work. And for episodic memories, maybe we want to look at the posterior parietal cortex to look at the integration of visual and somatosensory information, other attentional information there, and assign some activity to that. And for the visual spatial sketchpad, maybe we want to look at the visual cortex and the occipital lobe. And there's some reason we might think that this has some validity, because if we want to do something like fMRI task where someone is engaged in a visual imagery task, we might see some activation in the occipital lobe of the brain, and that would suggest that okay, when a task involves the visual spatial sketchpad, we see the occipital lobe become active. And that would suggest then that the occipital lobe is not just about what we're looking at right now, but any kind of visual imagery would seem to be associated with the activation of this part of the brain. I want to emphasize that this map on the right here is a little bit speculative, right? So we have reason to believe that these different brain regions play an important role in those different components of working memory, but I don't want to maybe be too beholden to saying this is exactly how it maps onto the brain. But nevertheless, a good lesson from this is about this equipotentiality business, right? It means that that for a task that is more visual or spatial in nature, then those visual areas of the brain are going to be more important and more contribute more to the performance of that task compared to, say, the linguistic parts of the brain. And likewise, for a task that is more verbal, then those linguistic regions of the brain will contribute more and visual areas will contribute less. So we see here that, yes, different parts of the brain are differentially more important. It is very much dependent on the nature of the task that we are performing here, right? So task dependency is pretty critical here. Let's say a little bit more about the role of the prefrontal cortex, or PFC. So we want to talk about the delayed response task. The delayed response task, that's a very broad term, by the way, but it's any kind of a task that is really a test of short-term memory in a way. So the idea is that we're going to give you some information, and you're going to have to respond to that information, but we're going to insert a delay so that it's not an immediate response. And so the critical point here is that as a function of this delay, you're going to have to retain that information for some somewhat short period of time. How do we distinguish between a short versus a long period of time here is a bit up in the air, but just something as simple as if I were to give you a series of numbers, and then after presenting you with those numbers, you have to wait 30 seconds before writing them down. That's a delayed response task, right? So you get the information, you got to wait 30 seconds, that's your delay, and then you respond by writing the numbers down. That could be done in a psych experiment, but more likely we would see something like what's called the delayed match to sample. So what usually happens here is that you see a stimulus on a screen. It could be something like a shape, like a red triangle, and then it goes away. And then after some period of time goes by, you see many shapes appear on screen. And one of them is the red triangle, but you could have a blue circle and a green square and various other shapes and colors. And you simply have to maybe point and click onto the item that matches what you had previously seen, right? So you have to match it, right? Or there could be a spatial version of this. So here, what happens is that you're looking at a fixation point on a screen, and a dot appears somewhere on the screen, and it appears for just a second, and then it goes away. And then some period of time goes by, and then you hear a beep. And your task when you hear the beep is to perform an eye movement to move your eyes to that same location on the screen where the dot had been. So you have to have a spatial memory of where that stimulus had appeared. And so this is a good kind of spatial short-term memory task, right? As opposed to the more explicit, I saw a red triangle shape, right? So any one of these different kinds of tasks could all be a delayed response task. Some could be more verbal, some could be more visual, some could be more spatial, right? What we see, though, is that in any of these cases, we see activation in the PFC. So if we're doing fMRI or FNIRS or any of these methods we've talked about, we can see PFC activation during delayed response task here. It's also worth pointing out here what we've talked about aging already. So another way to think about it is that separate from these imaging studies, what we can find is that for people who are aging, there's going to be a decline in the activity in the prefrontal cortex, and that also correlates with declines in their performance on a delayed response task. And so once again, we can say that, okay, not only do we see activation during the task, but in situations where there's a decline in that part of the brain, we see a decline in the task. As I had mentioned with the hippocampus, aerobic exercise tends to improve the hippocampus, increase the size of it, and that improves memory performance. Exercise also improves connectivity in the PFC. So once again, it's going to improve working memory performance in this particular case. True for older adults, It's also true for children and young adults. So there's been a handful of studies come out. I saw one come out just a couple of days ago, uh, where they did an exercise intervention in the classroom. Right. So with children, they had them learning, and then they do some exercise and test them. And kids in the exercise group did a whole lot better. So there seems to be some uh, argument to be made here that, like, say, a take-home message for you when you're studying, mix in a little bit of aerobic exercise before and after studying. It's very likely to have a solid payoff here. What else can we say about locations of memory in the brain? Well, let's revisit Lashley, because remember, Lashley was doing not delayed response tasks or anything like that, or complex motor skills like the mirror drawing task we did with HM. Lashley was doing conditioned eye blink responses, right? And so one way to do this in rabbits, for example, is you blow a puff of air into the rabbit's eye, and they're going to blink. But if you play a tone along with that puff of air, then the tone begins as a neutral stimulus, but eventually it becomes a conditioned stimulus, and rabbits will blink when they hear the tone. So we're going to train up our rabbits to do this, right? But what we're going to do is we're not going to just go in there and make a cut somewhere in their cortex the way Lashley was doing this. We're going to use something called a stereotaxic device. Right? What a stereotaxic device allows us to do is to insert a probe into a very, very specific region of the brainstem, the midbrain, or the cerebellum, and we can access a very, very precise, small little region of that part of the brain. And importantly, these are now subcortical areas. And what Lashley was doing, he was focusing on the cortex when he did his experiments. So, and he didn't have the technology of a stereotaxic device to be able to get to these very precise locations. But what we can do is we can not necessarily lesion, but we're going to suppress. And so this can be suppression can be achieved by using certain chemicals that essentially prevent those neurons from activating, so we can release a small amount of that into this part of the brain, and it prevents neurons from firing until that wears off, right? So now what we're going to do is we're going to train up our rabbits to blink to a tone. And now that they have been, uh, um, well, actually, hold on that, right? So we're not going to train them yet, right? We're going to suppress what's known as the lateral interpositus nucleus, which is in the cerebellum. And now we're going to start trying to train them, right? We're going to present the air puff and the tone, and we're going to try to train them up to see if they can learn to blink just to the tone. And while the lateral interpositus nucleus remains suppressed, we do this, right? And now once the suppression wears off, we're going to wait to see, okay, it's worn off. Do they show any sign of having acquired a conditioned response? And the answer is no, right? So during the entire set of trials, while the suppression was active, we don't see any signs of learning. And when the suppression wears off, we still don't see any signs of learning. And so let's start over now that the suppression has worn off. Can we train them up now? Yes, we can, right? So the entire time that we are suppressing this part of the brain, they seem unable to learn. Let's try it again. But now we're going to suppress the red nucleus, right? So again, we suppress the red nucleus and we start training them. And once again, we're noticing that they don't show any signs of exhibiting a conditioned response. They are not blinking to the tone. But now what happens is that once the suppression wears off, they start blinking to the tone. So, like, what's happening here, right? If we suppress the lateral interpositus nucleus, they don't appear to be learning. And once the suppression wears off, there's no sign that they ever learned anything. But when we do the exact same experiment with the red nucleus, at the beginning, it looks exactly the same. They don't show any signs of learning, but once the suppression wears off, now they do show signs that they were learning something the whole entire time. What does this tell us? What it tells us is that the lateral interpositus nucleus is important for acquiring the learning response. And when we suppress that, they simply cannot acquire the response. For the other version of the experiment where the red nucleus of the midbrain is being suppressed, because the lateral interpositus nucleus is functioning normally, the animal can acquire the response, but the job of the red nucleus is to initiate or activate the response. And they're not able to actually perform the conditioned response while the red nucleus is suppressed. So even though they're learning, they're forming the association between the conditioned stimulus and the unconditioned stimulus during this trial, the red nucleus is preventing the conditioned response from becoming activated. So once the suppression wears off, now they show a conditioned response because they were in fact learning, they just couldn't respond. So what in sum, this tells us then that Lashley might have been successful in finding an engram if he had been able to do this kind of an experiment, right? And this occurs a few decades later. So we'll say that the lateral interpositus nucleus is, in fact, a pretty important area of the brain for this kind of association, conditional learning. And the red nucleus is important for conditioned responses. This would then tell us that memories are stored in the brain. But once again, though, as I had said before, that they're not necessarily stored in a specific engram, right? A specific cell somewhere, right? So we've said before, for example, when we talked about the visual centers of the brain, there's no such thing as a grandmother cell, right? There's no cell somewhere in the inferior temporal lobe where all of that visual activity that when you see your grandmother's face, all of that visual activity might theoretically converge through a series of simple cells and complex cells all the way to some single cell somewhere that corresponds to the cell that gets activated every time you're looking at your grandmother. So that's what we call the grandmother cell, and we've rejected this possibility now. So we're not going to suggest that memories are stored specifically in one cell, but maybe we still want to talk about what we could call a network of cells, an assembly of cells, right? And so now we're going to talk about a distributed pattern of activity across a large number of cells here. It could be thousands, millions of cells even. And so what's happening is that across this entire network of cells, there's going to be changes that happen that are either going to strengthen some of the connections, the synapses, or maybe even weaken the synapses between neurons. And it's this pattern of strengthening and weakening of neurons, or synapses, sorry, that changes the activity of this entire cell assembly. And but that means now that what we need to do is we need to get into the details of what's happening at the cellular level. But what's important to keep in mind here is that if we have this entire neural network now that becomes active for a certain memory, that neural network is not just a neural network for one single memory, right? But rather what's happening is that we could create many different patterns of activation that spread across this network. And that means each individual neuron is not just a neuron for one memory, but it's participating in many different memories. So its role is not about remembering something. Its role is just to contribute to the overall activational structure of this network. And that can look different for different memory types. But again, as noted here, we have to get into the details of what's happening at the synaptic level here, right? What's going on with individual neurons and the connections between them, and how does that change? And how can we either increase or decrease the strength of a synapse? That, of course, is Hebb's law. And so that means that this is where we're going to get into next. But let's save this for our third and I hope final video for this particular chapter on learning and memory. And we will get into the famous studies next of aplesia, the sea slug, and what's happening there with the changes in the synapse, and then work out the details of long-term potentiation after that. So that will be it for this video, and I'll see you in the next one.